Okay, so we are live. It looks like everyone has been pulled into the room. Let me call Maurizio back on stage. And then I will fade into the background.
Okay, are you able to see me?
Yep, we've got you, Maurizio.
Fantastic. Sorry, I figured it out. I was in the other room. Okay. So I will quickly introduce everyone. The first speaker of the synaptic physiology panel. I'm very glad to introduce Richard Naud from the Department of Cellular and Molecular Biology, Medicine, at the University of Ottawa in Canada. And Richard is basically a fantastic speaker and also a great colleague. He did his PhD at McGill University, and then he moved. He studied physics at McGill University, and then he moved to do his PhD in Switzerland at Wolfram Gerstner lab in PFL. And then he did his postdoctoral training first in Cambridge and then later on in Berlin. So Richard contributed to the field on several topics. First on single neuron models, and then more lately on information processing capabilities and cortical neural networks. And today is actually presenting us some work also is doing on estimation of synaptic mechanism of short-term plasticity. Richard, please, the stage is yours, and good luck.
Thank you, Maurizio. Well, thanks to you, Maurizio, and all the organizers for this great event. I'm really happy it's taking place. It's very promising, so I'm really happy to get to see how it's going to develop in the next few days. And before I formally begin, I just want to encourage everyone to, interrupt me using the chat and ask Scott to interrupt me if you have any questions or anything. I like to feel like there's, I think someone's mic is not, yeah, thank you. I like to feel like I'm not talking to an empty room and that you're there. So, all right, now this talk, the premises of this talk is really actually starting from deep learning. The fact that deep learning can now take an image of an elephant and just say there's an elephant in that image is, although it seems very simple to us, it's such an accomplishment. It's such I didn't think when I was growing up that I would get to see a day that we would solve that problem. That has been obsessing philosophers for thousands of years. How do you get to know what you see and what's around you? And one big lesson that I get from that is that it is a switch from focusing on computation to focusing on the coordination of learning. I used to be focusing on computation, thinking that we would find computational capabilities of neurons and networks of neurons are so special, and that the brain has are so special they will give us, its formidable power. But deep learning has showed us that you can use very simple computational device. You just connect them together, and as long as you know how to play around with this, and in particular to coordinate what would think to be a synaptic plasticity, coordinate how the interconnections are set, then you're able to achieve formidable things. So that's the question I like to bring to it's a question I've been animating a new chapter in my research. And the way to frame it maybe would look like this, and for this talk, would look like this, where information flowing from bottom to top in this image here across a network of neurons. And at higher order areas where you would have the thing like elephant recognition arising. In lower order areas, it's more like pixels flashing up every neuron encoding the small parts of the of the visual field. If you're in vision, it could be other elements of smaller elements of sensory information. Then, if you're trying to set out those synapses that are in the middle, you ask the question: how would I choose whether this synapse should engage into LTP potentiation or LTD? How can I have this setting of this synapse depend on some type of control that is happening higher in the hierarchy? That's really what happens in deep learning. That's the coordination of plasticity. How can I have some control? Say I'm making an error in recognizing an elephant. How can I choose those synapses to appropriately change in order to optimize the function downstream? So that's the very general credit assignment problem. And today, I basically want to talk about one theory of how the nervous system can be doing this. And then in part two, which is maybe a smaller part, I'll talk about efforts that we've done, computational tools that we've done in order to help proving elements of that theory. And you will see it focuses a lot on properties of short-term plasticity. And really, in the part one is separated in three subparts. The credit assignment problem we think can be solved by three computational abilities. One is the ability of steering the plasticity at synapses through bursts. The other is a computational ability of multiplexing. And the last one is the alignment of the connections that are feeding back to the connections that are feeding up. And this part one is really a big collaboration between U Ottawa and McGill and Friedrich Miescher Institute in Basel. Blake and I are co-PIs on this, Blake Richards from McGill and Mila, and Jordan Gurdjieff and Alexandre Payer Alexandre is a postdoc and Jordan is a finishing PhD student that work together on working on all of this. And I should also point out that the basic idea of what I'm going to show today is coming from Koerding and Koenig in 2001, where and people have taken that idea back since Blake did that paper in eLife in 2016, where we basically now we're making that more and more precise and supporting that more and more with experimental evidence. So, in terms of synaptic plasticity, we would like to find a way how synaptic plasticity can be steered so that in the end I can have some type of controller that will be able to control that synaptic plasticity. So what are the main controllers, the main correlates of synaptic plasticity? I like to separate three main ones. One is relative timing. So this is the realm of spike timing dependent plasticity, where if you have a pattern of pre and post activity, when pre precedes post, you have potentiation, weight change that is positive, and when post precedes pre, in that case here, in this case here, you have depression, and that's relative timing. Then there's neuromodulation, where whereby different cocktails of neuromodulators at the synapse, around the synapse, that might be released by different centers, neuromodulatory systems, acetylcholine, serotonin, norepinephrine, will change the net sign and amplitude of the plasticity that you get. So you could change the cocktail, the components of the cocktail of neuromodulator to switch from LTP to LTD. And the last one is the firing patterns themselves. So if you pair a single spike with another single spike, you tend to have LTD. And when you pair a single spike with a burst, so the firing pattern of the postsynaptic cell in that example is changed, you tend to have LTP. And so these are three big elements are shaping synaptic plasticity. And today, I want to focus only on that last one. There's been a lot of work on those two first ones, relative timing and neuromodulation. But today, I want to focus on that last one and try to see how far we can get. What can this go? What can this do for us? So I'll build a simple model of this synaptic learning rule, and then I'll gradually add details in order to build a network that can learn. So first, I need to have a vocabulary to talk about these firing patterns. And I know this vocabulary is going to be too simple, but I need to have something to put words on. Most probably the real biology is going to be more complex, but I need vocabulary. So I separate a singlet from a burst simply by whether they have a relatively high firing frequency. So high frequency successive action potential will be considered a burst. And this is one burst, this is one singlet. And either one of those I will call events. This is my vocabulary. And then with using that vocabulary, if I pair a presynaptic event with a postsynaptic singlet, experiments tell me that I should get LTD. And if I pair a presynaptic event with a postsynaptic burst, experiments would tell me that I should get LTP. So this is Letzke's 2006. It's been reproduced by a few other labs. This does depend a little bit on a few parameters like development, and it only looks like this later in development. So this is my synaptic learning rule that I want to be able to put in a network and see whether I can use that to control synaptic plasticity. But typically, what you do in that sort of theoretical investigation is that you will control the presynaptic and postsynaptic firing rate, imagining that this is corresponding to information flowing in the network. But this doesn't work for when you're interested in finding out the role of spike timing patterns. Firing rate would have only one knob and doesn't take into account the fact that this is not the same spike timing pattern as this. This got the same firing rate. So instead, I need something that has two knobs, some type of model of firing that has two knobs, and I do that with the event rate and the burst probability. So the burst probability is simply the fraction of events that are burst. So for instance, here I have 4 events, and here I have 4 events. Here, there's no burst, so the burst probability is 0. And here, there's 1 burst out of 2 events. Sorry, I said there was 4 events. There are 2 events here. So there's 1 that is a burst, 1 that is a singlet. So it's 2 events with a burst probability of 1/2. And this allows me to have 2 knobs that I can change fairly independently. The burst rate then becomes just the event rate times the burst probability. And then I can study my synaptic learning rule. I can create a presynaptic and a postsynaptic neuron that fire synchronously with a predefined event rate, pre and post. And I can vary the burst probability, keeping the event rate fixed. And if I simulate this sort of learning rule, you'll see that as I increase the burst probability, I will get into a LTP-dominated regime. This is natural. I mean, when you increase the burst probability, you have more of those LTP eliciting pairings. And if you have a low burst probability, you have more of those singlet pairings, and you have more LTD. And so this burst probability allows you to control the plasticity. You can change the event rate. So if, for instance, I go instead from 10 hertz to 5 hertz, the event rate is changing here, it's reduced. I change effectively the slope of that curve, but I don't change the fixed point, the point where it switches from positive to negative. And the rule mathematically would look like this. You have event rate pre times event rate post. That is multiplied by the burst fraction minus the average burst fraction here. This is at this point, f bar. And that is a signed quantity that can be positive or negative depending on the burst fraction, the burst probability. And all this together is setting out the weight. And you see, therefore, there's something like, this is really like a Hebbian term. Theorists would call that a Hebbian term. And this allows you to steer plasticity, changing the sign of plasticity. So cool. I have a mechanism that is able to, if I'm able to control burst probability, I'd be able to control whether the synapse is engaging in LTD or in LTP. That's a pretty cool thing. But in self, it's not doing much because I'm not able necessarily to control burst probability. How can I control burst probability? Well, this is where dendrites come in, we think. The blueprint of the cellular level network connectivity across multiple columns throughout cortex is such that you have mainly bottom-up, means sensory, if you're in sensory area, information is going to the basal dendrites of the large pyramidal cells of the cortex. And layer 1 receives feedback or mostly top-down information that we're not quite sure what it really conveys. It can be some type of attention, expectation, learning signal, coordination between different areas. But the fact that it could be a learning signal, something that made us think, all right, what if a dendritic input comes in here, and we know that these sort of dendritic input and those thick, tufted, deep layer pyramidal cells are able to control bursting. So this is a classical experiment by Matthew Larcombe in 1999, where they've forced a spike in the soma that is seen here. And they played with the timing of the input that was injected in the apical tuft here, the bifurcation of the apical tuft. And you see here, this is the EPSP-like injection that is combined with the backpropagating action potential. Now, if you play with the timing of that input, you suddenly depolarize the membrane longer here that allows some calcium ion channels to generate this large and very important event, the calcium spike, that lets a lot of calcium in and forward propagates to elicit a burst. And if the timing is just a little bit off, you come back to a single spike. So effectively, what happened here is with properly timed and properly sized dendritic input, I was able to switch a singlet from a singlet to a burst, meaning that I was controlling burst probability. I didn't change the number of events according to my vocabulary here. I only changed the burst probability. So input here in the apical dendrites would allow us to take that knob and use it to control burst probability, and input here in the somatic area, the proximal area, would allow me to control the event rate, the net number of singlets or bursts that are occurring. Okay, let me just illustrate how this could work in a simulation. Maybe that will help you visualize. I have a simulation here where a presynaptic cell that receives synchronous inputs is sending one synapse to a postsynaptic cell here, which is receiving synchronous input here, but I will control the net amount of distal input, the input to the apical dendrite. And what I'll show you is the weight mainly. So this is the synaptic weight. And I want to be able to control that synaptic weight by controlling the currents, the amount of input that is coming here. So that I have this controller. And I'll show you here the presynaptic burst probability and the presynaptic event rate that remains constant. And here I will basically, when I have a strong distal input or weak distal input, I should have the burst probability of that ensemble that is shown here with the red line and the axis on the right will increase because I have this calcium spike generating mechanism in my model here. And if I have a weak distal input, it will decrease. And if I'm able to control that synapse, well, the weight should follow. And this is what it looks like when you do the simulation. So by injecting a current here, I'm able to control the plasticity of that synapse. Effectively, like biophysically, although this is a model, this means that by allowing to control the generation of calcium spike, you're able to control bursting, which means that you're able to control the net depolarization in the basal dendrites where the synapses here are, and you're therefore able to control part of the synaptic plasticity if things are working this way. So all this to say that I'm able to put my controller, some types of synapses, some number of synapses away as long as they are controlling the net input in that dendrite. But now, if you want to use that to control plasticity, there's another problem. And I'm getting to my next problem, part 1.2. There's another problem that arises because when you're changing the burst probability, you're changing the burst rate, meaning you change the net number of action potentials that are being produced by this neuron. And therefore, when you're trying to communicate that information up the hierarchy, you've changed that information according to something that is a learning signal. So if I see this steering signal as a learning signal, it should be distinct from the information that is carrying the sensory information or other information that you want to use to do computation. And you might want to actually use that learning signal to send it back down in a way that will simulate the backpropagation of error algorithm. But again, it should not be polluted by the sensory signals, at least not if it's in a particular way. It needs to be done in a particular way to backpropagate a credit signal. So how can that work? How can you have two signals, meaning different things, being propagated in different directions in the cortical hierarchy? One that would, if one is sensory information, one could be inherited credit. And down here, if you're following the backpropagation of error algorithm, it should be a unit-specific credit that's something like the sensory information times the inherited credit. So the solution to this, as I hinted at in my introduction, would be multiplexing and the presence of polarized short-term plasticity across the cortical hierarchy. So burst ensemble multiplexing is a coding ability that I discovered with Henning Spreckler working on in my postdoc. And I try to illustrate how it works here. So normally, if you look at ensemble coding, where you have an input that's injected in a population of cells, cell 1, 2, 3. This is the output of cell 1, output of cell 2, the output of cell 3. And you would then look at the bin per bin ensemble firing rate by counting the number of spikes that are in every bin. And you see, there's 3 spikes here, there's 3 spikes here, 4 spikes here. And the ensemble firing rate is slowly increasing, reflecting the input that this ensemble received. Well, this is the ensemble rate picture. It's a classical picture. But if you think that firing patterns are meaning something different, then you should maybe separate them and calculate the bin per bin, the number of the different firing patterns that you have. So for instance, here, if I use my vocabulary, I have one event that is a burst. Here there's one event. So if I look at the event rate, there's one event in that time bin, there's two events in that time bin, and there's three events, 1, 2, 3 in that time bin. I don't count the second spike because this is one burst, it's one event. And see, you have a strongly increasing event rate through time. That might have been caused by somatic inputs strongly increasing through time. If I can look then at another feature of that spike timing code, which would be the burst probability. Here, the burst probability is 1. There's one event that is a burst. Here, the burst probability is 1/2, and here is 1/3. So it's decreasing in time, meaning that I have this other ensemble rate feature that is completely independent from this one. And that might be reflecting the dendritic input. So if I have something that is able to route based on that classification of firing patterns, the information, I might be able to carry to different parts of the cortex learning signals and sensory or computation relevant information. And it turns out that short-term plasticity would be the best mechanism for this. Quite obviously, and in fact, this is really where my vocabulary comes from. Because many synapses, many neurons, when they send axon, depending on the postsynaptic target, will make either a short-term facilitating synapse or a short-term depressing synapse. A short-term facilitating synapse would look like this, where if you have a burst, the first spike, which alone would be the singlet, really doesn't do anything on the postsynaptic membrane. But only if you have a long succession of high-frequency action potentials do you actually have a strong effect on the postsynaptic membrane. So one can simplify this and looking at it like this, where if you have a singlet, it just doesn't communicate anything. And if you have a burst, it will communicate something. So it will make an EPSP. And conversely, short-term depression is one where the first spike, the event, whether it's a singlet or a burst, first spike is going to be communicated well. But the later spikes in high-frequency bursts are attenuated. You see the EPSP is very small here, meaning that I can simplify it like this, where a single spike will make a big EPSP, but a burst will just make the same big EPSP as if there were a single spike. The other 2 or 3 spikes are being attenuated so much that they don't really make, they don't really have an influence. So this would allow me to route different types of information. So let me illustrate the multiplexing in another simulation where I've injected that sinusoidal current at that ensemble here that receives it in its basal dendrites or soma, somatic compartment. It sends an axon to this pink population here, and it's making short-term depressing synapses both in the main cell body and in a feed-forward inhibition model. Then I can compute the event rate of that green population, and of course it follows a sinusoid that I've injected. And I can compute the event rate of that pink population, and of course as a consequence, it follows really well the sinusoid with a little bit of a phase shift that is due to the spike frequency adaptation that those models have. But what I haven't told you is that in that same simulation, I was also, during all that same time, injecting a current in the apical dendrite that was a slower varying sinusoid. And that slower varying sinusoid that is injected in those cells is not affecting the event rate at all in neither of those cells, even though there was actually a feedback connection to the apical dendrite that was using short-term facilitation. It's just not there in the event rate. It's not there. Why? Because it only affects the burst probability and not the event rate. So if I plot the burst probability of this pink population, you see it follows perfectly the burst probability, the input that was injected. And now if I can look at the burst probability of that green ensemble, now what should that be? Well, if I have, what comes out of this will be something that has an event rate that's modulated by this burst probability. The burst rate is a multiplication between the two. Then this is the burst rate of that pink population that will be selected for by my short-term facilitating synapse and will therefore modulate my burst probability here. And this is exactly the green, the orange here is the burst rate of that ensemble, and the red here is the burst probability of that ensemble. So you see that I've communicated two signals in different directions. One is just unaffected by the one going up is unaffected by the one going down, and the one going down is affected by the one going up, but only in that multiplication. As I hinted to earlier, this is along the lines of what you would need to do to implement a backpropagation of error algorithm.
Richard, I'm sorry.
Yeah.
You have still 5 minutes.
Okay. Yeah, thank you. Thanks, Scott. So I'll wrap this whole story up quickly, meaning so with this sort of circuitry, we built a rate model that emulated the main dynamics and showed that we can, on a very difficult task, ImageNet, learn to classify those millions of images. So we built that 6 convolutional layers with a few fully connected one fully connected layer, and we show that well, if we keep our weights on the feedback fixed, we are not able to learn. But if we have a learning rule that depends on the bursting again in the feedback as well, which is this line here, we're able to learn to recognize those images in a way that doesn't really match exactly backprop. We have some reasons for that, but it's certainly learning much more than, say, what you would achieve with Naud perturbation, as is shown here. So this is ImageNet and this is CFAR 10, another training set. And the summary of that part 1 is that supervised learning, that burst timing plasticity, this third factor in regulating synaptic plasticity, can help you do some type of supervised learning where you're able to controlling or coordinating the plasticity across ensembles. Now, the main prediction that I find in this, one important prediction is that bursting should be related to learning. We have made some efforts to address this, and please see the trainee talk by Zach Friedenberger on that topic. But the other one is structural, is the fact that you should have some type of polarization of short-term plasticity across the ensemble, such that when you're going down the hierarchy, you should send a short-term facilitating, and when you're going up the hierarchy, you should make short-term depression. And very shortly, we've made a few efforts, and since I'm not an experimentalist, I'm not able to do that myself, but I can create tools that will help experimentalists to measure this, which has been hard to come by. There's 2 observations, one in the thalamus. I'm just going to skip that to go straight to our results, and skip this one too, on the observation hours in cortex. Because they are not able to pinpoint at specifically intercortical areas, whether there is short-term depression and short-term plasticity. So we created 2 tools. One that is applicable to the glutamate release sensors. So collaborators use this glutamate sniffer, fluoresces in the presence of glutamate, where you see in a single synapse the amount of glutamate that's being released. So here there's a stimulation, and here there's a bit of fluorescence that relates to the amount of glutamate that's being released. And we built a model of what will be possibly multivesicular release and we fit that model using expectation maximization algorithm, it's able to give us the properties of the synaptic release, the number of vesicles, the parameters of their size, and the probability of release of the individual vesicles. And we found in those experiments, and it's in that paper here, that the central synapses are releasing on average 2 vesicles per synapse. And then the other tool that we developed was to capture the vast number of different types of short-term plasticity that you see: facilitation, depression, delayed facilitation, biphasic, where you have facilitation followed by depression. And we built a model that basically is encapsulating all of these different modes into the shape of a kernel that is convoluting the input spike train, and then is read out through a nonlinearity before being sampled to generate the postsynaptic amplitude. And with this, we were able to show that our model is outperforming the standard models, the Zodix micron model, and the fact that we're able to infer the properties of that model efficiently makes me hopeful that we'll be able to use that model to characterize short-term plasticity in cortical networks. So with this, I'd like to summarize what I've said to you today, that bursting can solve the credit assignment problem if you allow burst-dependent plasticity and polarized short-term plasticity in cortical networks. Then we have this characterization method to infer the properties of single synapse release, and we have this model that can be inferred on plasticity protocols to characterize the short-term plasticity of synapses efficiently. So with this, I'd like to thank again my collaborators for the first part, my experimental collaborators, funding for making this work available, members of the lab. Dan Trotter made the single synapse algorithm. Julian Rosperich made the short-term plasticity algorithm. And I'd like to highlight that I have open postdoc and PhD positions that are open. So if you're interested, please contact me.
Thank you, Richard. And I'm afraid that we are a little bit off the clock. So I would like to ask the audience to retain the questions, and hopefully Richard and all the others, including myself, will address them at the panel. And without further ado, I thank you again, Richard, and I would leave the stage for introducing Gianluigi Mongillo, who is the next speaker. Thank you.